Welcome again. Today we are going to look at the synthesis of fatty acids. Last time we looked at the beta oxidation of fatty acids. Today is the synthesis. Here. So fatty acids are usually synthesized by an enzyme known as fatty acid synthase. But this fatty acid synthase is a complex of 6 enzymes which are linked to an acyl carrier protein, which is found at the center there, which is also linked to 4' phosphopanthine, a derivative of vitamin B5, pantothenic acid. Should look at that probably. So let's start here. Yes. So the first enzyme is ACP. When I say ACP, I mean acyl carrier protein, like this one, acyl carrier protein. So ACP, the first enzyme in this reaction, essentially in this reaction and this reaction, this ignore whatever we have carried out here, ignore it. So the first enzyme is an ACP acetyl transferase, which usually transfers this acetyl CoA here and links it to this SH group. This SH group here, a thiol group, which is linked to a cysteine residue. The cysteine residue is found on the beta ketoacyl. The beta ketoacyl synthase. So the cysteine residue is bound to the beta ketoacyl synthase is a residue. Remember, these enzymes are simply proteins in nature. Proteins are made up of amino acids. Cysteine is an amino acid. If you can remember the structure of cysteine, it had a thiol group. So this thiol group. So acetyl is linked there, just as shown here. It's linked there by this enzyme here. Maybe I should write that for you. So the enzyme here is ACP acetyl transferase. Yes, that is the enzyme. I should bring it here. No, stay there. So that is the enzyme responsible for that reaction. So then the second reaction. So the second reaction here, we add a malonyl. So you know a malonyl? This is the structure of a malonyl CoA. How is malonyl CoA formed? I will show you later. But simply we carboxylate an acetyl CoA. See this one in blue is a CO2, which was a CO2 group, which was added by acetyl CoA carboxylase enzyme. There, remember the coenzyme is biotin. Biotin is the one which carries. I will show you the reaction later. So here in this reaction, via an enzyme which is known as ACP malonyl transferase, we add a malonyl group here to this SH, which is linked to the acyl carrier protein. Okay, so we add this malonyl group, and we use maybe another color. This malonyl group here, we added here. Okay, so we get this. Can you see how it was added here? So once we do this to the system, it's like we have loaded the fatty acid synthase. Okay, we have loaded it with acetyl and malonyl CoA. So after loading, we will carry out 4 reactions. 4 reactions are carried out. First one is condensation. Second one is fast reduction. Third one is dehydration. Fourth one is not the fourth, sorry, is the second reduction. Remember in the beta oxidation how they were the sequence. In beta oxidation, it's fast dehydrogenation. Then we had a hydration, not dehydration. Then we had the second dehydrogenation, and then we had the cleavage step. If you haven't seen the video on beta oxidation, you shouldn't be here. Go and first look at it. So here I've said it's we have said condensation, fast reduction, dehydration, second reduction. So this is the condensation reaction. Yeah, can you see what I want you to see? The reaction mechanism. Let me clear all this, but before I clear it, let me clear. Yeah. Yes, I want you to see this. Can you look here? Not there, sorry. Can you look here to this here? I want you to look there and there. Why are we calling it a condensation step? First, we will cleave this bond, the one pointed here, and then we will cleave also. Those 2 bonds will be cleaved, okay? After cleaving, this group in red is transferred there. And the other one in blue, the CO2 is the one lost, okay? I hope you have seen the reaction mechanism. We cleave this bond between the where we added the acetyl, then we added to here we have removed the CO2. Okay, so we make this new bond here, and CO2 is lost. So this reaction, that reaction is usually catalyzed by another enzyme, which is known as this compound now we have formed is a ketoacyl. So that enzyme, that is why it is known as a beta or a 3 ketoacyl synthase. Beta or a 3 ketoacyl synthase. Synthase, since it is synthesizing a ketoacyl, a 3 ketoacyl synthase. Let me move it a little bit there. Yes. Okay. You have done that. So I hope you have seen the reaction mechanism. Let me repeat. We cleave this bond. Let me do it in another color. There is no harm. We cleave this bond, and also this CO2 is lost as shown here. Then this group in red is added there where we have removed the CO2 to make this new bond. Now this compound formed is since this is we have carbon number 1, carbon number 2, carbon number 3, or the beta carbon. So this is a 3 ketoacyl. Since it is linked to an ACP, it is let's call it a 3 ketoacyl ACP. It's linked to an ACP, as you can see. This is an ACP here. So this enzyme is you can either call it 3 ketoacyl synthase or a beta ketoacyl synthase. This is the condensation reaction. Let me in fact, I had forgotten to write that. Let me write that. This is a condensation. Condensation. Okay, this is the condensation. Okay, then the next reaction will be the fast reduction. Fast reduction. Why? What is reduction from your chemistry? Reduction can be defined in two ways. Now, three. First reduction I know is removal of oxygen. Two, reduction is the gain of electrons, or reduction is the gain of also hydrogen. Okay. So let me show you why are you calling it reduction. So here we bring in nicotinamide adenine dinucleotide phosphate, which is reduced, since it has we have this hydrogen there and there. Okay, so these hydrogens are the one which we will add. Can you note how they have their color? They have somehow. So where shall we add them? Let me clear so that I can show you where we will add them clearly. This is added here to form an OH and shown here. Okay, then this is added here. Okay, to form there, as you can see. Yeah, so I said this is the you have to repeat that. This is the first. This is the first. What am I writing? This is the first reduction. And I've shown you why I calling it reduction. This is the first reduction. And it is usually catalyzed by an enzyme. Since we are reducing a beta ketoacyl, so what do you expect the name to be? Yes? Did I hear you say beta ketoacyl reductase? That is correct. So beta ketoacyl ACP reductase. Remember that it is linked to ACP. Or I can call it a 3 ketoacyl reductase. Okay, that is the enzyme responsible for that. I hope you have seen the reaction mechanism. It's not that hard. Then here we carry out dehydration. What is dehydration? Dehydration, move of water. So here is where we are carrying out the dehydration step. Dehydration. So we are carrying out the dehydration step. So let's name this compound we have formed, this one. What is the name of this compound? So first it is ACP linked, then this is carbon number. Let me do that. Sorry. This is carbon number 1, carbonate carbon. Number 2, then, so at carbon number 3, we have an OH group. So it can be 3 hydroxy. Then this is which configuration? The OH group is in the D configuration. So it will be a 3 hydroxy D butyryl ACP, or simply a beta D. Let's call it a beta D hydroxy acyl. Let's call it like that. A beta D hydroxy acyl, so that you can have an idea of the name of the enzyme. So what is the name of the enzyme? If you were to name it, how would you name it yourself? Yes? A beta if I was given the opportunity to name it, I would name it a beta or a 3 hydroxy. Let's call it a 3D hydroxyacyl hydroxyacyl ACP dehydratase. Okay, a dehydratase removes water, a hydratase adds water. So a dehydratase enzyme. Okay, maybe I should move it a little bit down. Okay, this is a dehydration step. We remove water, and the enzyme is a 3D hydroxyacyl ACP dehydratase, or simply can call it a beta hydroxyacyl ACP dehydratase. Okay, where now I want to show you which waters are we removing, if I can use that term. Which waters are we removing? Where are we deriving these elements of water, H and the OH? See, here we will remove this OH, and here we will remove one hydrogen. So a hydrogen plus an OH, H2O, your water there. Okay, so after removing, here you remain with one hydrogen. Remember, this hydrogen was the one which was derived from NADH, NADPH, and this other one. So here we have a double bond. So what is the name of this now new compound formed? This is carbon number 1, carbon number 2. So if I call it a trans, remember this hydrogen one is, they are in opposite, so it is a trans delta 2 butanol ACP, or simply call it a trans delta 2 enol ACP. Okay, a trans delta 2 enol ACP. Remember, ACP is the acid carrier protein. Then that was the dehydration step. Then let's go to the last step. The last step is the second dehydrogen, not the second reduction. Let me now clear all those here. Now this is the second reduction, just like the first reduction where we add in hydrogens from NADPH. So let me show you. Can see this hydrogen, this one, it is added here. Okay, and this other one is added here to form this. See, the double bond has disappeared. Now you have formed a single bond. Okay, so upon forming this, now this itself, you can see it has somewhat the structure of a fatty acid. This is the terminal carbon, that one. Then you can see here is we have a carboxyl carbon here. Okay, then we will carry out the rust reaction. Okay, the rust reaction will be the transfer. The rust reaction is simply a transfer. See, it was here down, here it is here. It is transferred from the SH group, which is linked to ACP, to the SH group linked to the cysteine. Remember the cysteine was linked to the beta keto acids in this. So it is transferred there like this now. Okay, so it has already been transferred. Now, in the corresponding reactions, we will only be adding malonyl CoA. We will be adding malonyl CoA. So, in the other reaction, the first reaction which we said of the ACP acetyl transferase is omitted, but the other ones are retained. So we will be adding a malonyl CoA. Then we carry out the 4 reactions: condensation, first reduction, dehydration, second reduction. Okay, like that. So we will be, for every cycle we carry out, we will be adding 2, 2, 2, 2, 2. Remember, although malonyl CoA has 3, remember we are losing a CO2 in the beta keto acids in this reaction. Okay, so let's say we are forming palmitic acid. So up to here we have 4. We have 4. Palmitic acid 16. So 16 minus 4, 12. 12 divided by 2, 6. So we will carry out further 6 more reactions. Let me show you. So here is another one. No, let's say we have carried out up to. So this will be, let's say, a palmitic. 5 more. Now let me come here. Yeah, in this reaction, I will show you how. Okay, let's do the second. Here we add a malonyl CoA via the ACP malonyl transferase. Then we carry out the condensation. We have removed. Then this is the first reduction number 5 here. This is the second one. And then not the second, dehydration. This is the second. So see, we have added 2. So initially they were 4 carbons, but here it is 1, 2, 3, 4, 5, 6. We have added 6. So let's do that. So it will be 6. Let's say we are forming palmitic acid. 16 minus 6, 10. 10 divided by 2. We need to carry out 5 more reactions like those ones to form palmitate here. Okay. Then remember, palmitate, we need it as a free fatty acid, not the one linked to the fatty acids in this. So via an enzyme known as the palmitoid thioesterase, there you add water, we cleave this bond here to give you your fatty acids in this system and your palmitate. Now the palmitate is the free fatty acid. Okay, so the reaction is simple as that. Let me show you how malonyl is formed very fast. I had an image here. Yeah. Let's go down here. So here you can see this is an acetyl CoA. 2 carbons, 1, 2. Then here this reaction is catalyzed by the acetyl CoA carboxylase, where you see a bicarbonate. This, we remove our CO2 from here, we add it here. We added to this side here to form the malonyl. This is the structure of malonyl CoA. This reaction requires ATP, by the way. So I want to show you something. That reaction is catalyzed by the acetyl. I think I'll say the acetyl CoA carboxylase. Acetyl CoA carboxylase is linked to biotin. Remember, biotin is vitamin B7. Yeah, B7. So how do you? This reaction is this reaction, acetyl CoA when inactive, so acetyl-CoA is an oligomeric protein. So the structural units of an oligomeric protein are known as protomers. So these are the protomers. These are the protomers. So during activation, what goes on? So citrate usually activate this acetyl-CoA. What does it do to activate it? Simple. It will simply link all this. It will make the oligomeric, the protomers to bind, to come together to form the oligomeric protein. So you see the first up there, they were scattered, if I can use that. Then here they are linked. So once linked, it is an active polymer. But palmitoyl-CoA, remember palmitoyl-CoA was being formed in the activation step of beta oxidation. You cannot be carrying out oxidation and at the same time you want to carry out synthesis. So those two pathways are antagonistic. So palmitoyl-CoA usually inhibit. So with that, I think I can say I've come to the end, and thank you.